Hello, and thank you for joining us today for the latest DDN webinar. My name is Marnie Willman, Assistant Science Editor, and I am here to moderate today's event. I'm very pleased to introduce Brian Haldeman. Brian Haldeman is the Product Manager for Leprechaun at Unchained Labs. Prior to this role, he was a field application scientist supporting biophysical characterization of proteins and viral vectors. Haldeman received his PhD in biochemistry from the University of Nevada, Reno, where he studied the mechanics of muscle contraction. His research utilized single molecule microscopy, pre-steady state kinetics, protein affinity and binding characterization, and protein isolation techniques. A very warm welcome to you, Brian. Following the presentation, we will have a Q&A session, and we welcome any questions you may have. Please ask questions using the Q&A system on the side of the live stream player. Without further ado, I will now hand over to Brian to begin their talk.
Great, thanks so much for the introduction. I'm really stoked to show you how Leprechaun gives you unmatched EV characterization. Before we get into EVs, I want to start with a brief history lesson from my home state of Nevada. Aside from gambling, a big driver of our economy is mining. These days, it's primarily rare earth minerals, but back during the gold rush, miners were looking for silver and gold. Along with these precious metals, the hills were also full of the mineral we see here on the right, pyrite, also known as fool's gold. Many inexperienced miners were convinced they'd struck it rich when they found pyrite due to its physical similarities to actual gold, shown on the left. Without an in-depth characterization, they were easily mistaken. Just like gold, extracellular vesicles can be hard to find and are commonly found with less valuable but similar looking particles. They're found in many sources, from cell culture media to biofluids, but are particularly challenging to characterize for several reasons. 1. Samples are complex. There's lots of other stuff in them besides EVs, for example lipoproteins, cell debris, protein aggregates, etc. You need to know that you're analyzing actual EVs and not other particles that may look similar. 2. EVs are really small. They're sub 200 nanometers. There's few optical techniques which can size down this small accurately. Next, they're super polydispersed and highly heterogeneous. You really need a single particle technique to get accurate information. Bulk analysis techniques, things like DLS, struggle to provide meaningful data when polydispersity is this high. And you're probably looking for a rare population. Not many EVs in your sample have your marker. In this case, you may not need to only identify EVs, but identify a rare subpopulation that contain your marker of interest. All this means you need a method which is sensitive, specific, and can size the small stuff. But even amongst the most recent single particle techniques, most struggle with the unique challenges of analyzing EVs. Sizing limits of these methods vary depending on the purity of the sample, but they all struggle to get down below 50 nanometers. They also have limited phenotyping capabilities, either due to photobleaching with NTA or a complete lack of phenotype analysis. Not to mention that EVs must be isolated to get meaningful data from any of these methods. The isolation process puts your sample at risk of losing these already rare subpopulations that you want to analyze. Basically, this means the quality of the data you get out is only as good as the purity of the sample that you put in using these techniques. Today, I'm going to introduce you to Leprechaun, the only tool that can fully characterize EVs from only a few microliters of unpurified, crude samples. Since we're doing history lessons today, I also want to cover the history of Leprechaun. The technology in Leprechaun came from a company called NanoView, which Unchained Labs acquired in 2022. NanoView was founded in 2015 and were the first to commercialize the core technology behind Leprechaun, known by the acronym SP-IRIS. NanoView released 2 generations of the ExoView instrument before being acquired. Leprechaun continues to build on that technology and can be viewed as the 3rd generation ExoView system with additional capabilities. In one single assay, Leprechaun can provide EV concentration, size, now down to 35 nanometers, and analyze expression of up to 4 different markers per EV. So you find out everything you need to know about your sample from a single assay. Even better, it does this on a single particle basis, without the need to purify your sample beforehand. Leprechaun characterizes different particles, including EVs, on up to 16 samples at a time. Add a maximum of 25 microliters of your sample to our Luni consumable, which will capture your EVs. Then run the Luni on Leprechaun, where it combines accurate sizing via interferometry with 3-channel immunofluorescence to get all the details on the structure of each captured EV. This image shown on the right is taken directly from the Leprechaun analysis software and shows individual EVs decorated with fluorescently tagged antibodies for 3 different markers. From just a few microliters of crude, unpurified sample, you can get all the key information on your EVs, including replicates, from just a single run. There's no need to spend time concentrating and isolating EVs from your sample. Whether it's cell culture fluid, whether it's biofluids, Leprechaun's compatible with them all. And with hands-off data acquisition, there's no need to spend hours manually loading and running your samples. Our newest Luni configuration, the pan-tetra Luni, is coated with a mixture of highly specific antibodies targeting the big 3 exosome tetraspanin markers, CD9, 63, and 81. These antibodies allow you to specifically capture EVs from even the crudest samples. Each Luni configuration features replicate spots for each tetraspanin capture probe, which grab hold of your EVs to ensure you're only quantifying true EVs and not those particles that may look similar. We always include an isotype control as well to ensure particles are specifically bound to the Luni surface. Once captured, you can go straight to sizing and particle count, or you can incubate with fluorescent antibodies to pull all the structural details about your EVs. Quantifying EVs is as simple as diluting your sample in the kit-provided buffers and adding a total of 50 microliters to the Luni surface. Zooming in on the Luni, you can see individual EVs binding to the surface of the capture spot. This specific antibody capture of EVs is what allows Leprechaun to work with crude samples. It pulls your particles of interest directly out of solution and binds them to the Luni surface. Once your sample is incubated on the Luni, all sample prep is handled by the Luni washer. From washing away non-specifically bound particles, staining with fluorescent antibodies, and drying to prep for imaging, the Luni washer handles all of this with preloaded protocols so that you can handle more important jobs in the lab. It'll let you know when it requires a manual intervention. From incubating your sample on the Luni surface to analyze your results only takes a few hours. Leprechaun gives you options on how you want to process your samples, from a simple size and count to give you EV titer, to addition of the optional kit-provided fluorescent antibodies to look at the tetraspanin profile in your sample, or you could choose your own fluorescent antibodies to identify those custom rare biomarkers within your total EV population. Leprechaun gives you the flexibility to design the assay around the answers that you want from your samples. Now we're going to dive into some data that you can get from Leprechaun. This first example shows a quick and simple experiment with no fluorescent antibodies to determine both EV titer and size. The images from Leprechaun show individual particles bound on the capture spots, which are then counted and sized on a single particle basis. This gives you EV titer in particles per mil and a size distribution of all captured particles from your sample, without the need for EV isolation or concentration. Stepping up the assay to get more details on your EVs includes 3-color fluorescence, which confirms which proteins are present on an EV, giving you structural information about your particles. In this example, the fluorescent antibodies for the big 3 tetraspanins were incubated with the samples. Leprechaun takes sequential images using the red, green, and blue fluorescent channels, and then overlays with the interferometric sizing channel. When the channels are overlaid, it shows which markers are present on each individual particle to determine subpopulations that contain combinations of all the different markers. For example, particles that are single positive for CD81 are shown in green, and dual positive for CD81 and CD63 shown as yellow, indicating overlaid green and red. Leprechaun can then automatically identify which particles are present in each channel to build up a picture of the structure of each individual particle. The output from this example experiment shows not only total EV titer indicated by the gray bar, but also the titer of EVs that are positive for each individual marker, shown by the red, green, and blue bars. Size profiles can also be broken down into each individual channel as well to give further insight as to whether particles with a specific marker tend to be larger or smaller than the rest of the population. As a proof of concept experiment, we took an EV standard isolated from HEC293 cells and spiked in a known amount of GFP-positive EVs sourced from Vesiculab. Using this experimental design, EVs from both populations could be identified using the red-labeled CD63 antibody, while the GFP-positive EV population could be identified from GFP fluorescence using our blue excitation channel. By combining sizing information with structural info from fluorescent imaging, Leprechaun can separate EVs into distinct populations. From the results, we can see that the Leprechaun analysis software was able to count total EVs, shown in gray, those that contain CD63, shown in red, which was almost all the EVs, and then the subpopulation that was positive for GFP, shown in blue. The Leprechaun analysis software takes that subpopulation analysis one step further and separates all EVs into populations that were either single positive, just for capture, and no fluorescence, dual positive for one of the fluorescent markers, or triple positive showing colocalization of both fluorescence channels with interferometry. The sizing profiles also show a distinct difference of EVs that are GFP positive. Their mean diameter, shown in purple, is shifted larger, indicating that EVs containing GFP cargo have a larger size, and that makes sense. Speaking of cargo, now is a good time to mention that Leprechaun fluorescence analysis is not limited to surface markers. By gently fixing and permeabilizing the EVs using kit-provided reagents, you can probe for internal cargo proteins alongside your surface markers. Here, we detected the internal protein syntenin alongside CD81 and CD9. Without permeabilizing the samples, detection was isolated to the surface markers, shown in the image here, as well as in the titer profile over here as well. When you did permeabilize the samples, this opened up the internal marker syntenin to our red fluorescent antibody, shown in the image, as well as in the titer profile here, where we see an increase in red fluorescence because the syntenin is available. Here's a great example of the power of EV cargo analysis with Leprechaun, published by one of our customers. Doxorubicin loading into EVs was measured using Leprechaun. On the left images, you can see that the overlay of fluorescent dox in green, as well as with CD63 in red. The bottom image shows the colocalization of the two, confirming effectively loaded EVs. Size analysis and the results show that dox-loaded EVs were about 10% larger than the empty EVs, shown by the size profiles and the determined mean size in the populations. This data was also used to measure loading efficiency, which is absolutely critical when designing engineered EVs to be used as a therapeutic. As I've said several times so far, one of the biggest benefits of Leprechaun compared to other technologies for characterizing EVs is the ability to work with crude samples. We've seen Leprechaun used with a variety of EV sources, including cell culture media and many different types of biofluids. The wide dynamic range of Leprechaun, from 5E5 to 5E8 particles per mil, allows these samples to be run with a simple dilution into our kit-provided incubation buffer without the need for isolation or concentration. We have established protocols with best practices on how to dilute each type of biofluid and how long to incubate to get you in the right ballpark for characterizing your EVs with minimal optimization required. If you want to customize the EV capture step to only analyze EVs with a specific marker, the Leprechaun Flex Kits are a great option. Flex Kits let you customize the capture step from the comfort of your own lab, with a few additional steps outlined here. You want to start with a well-performing antibody that has been validated using an orthogonal method, such as flow cytometry. Just incubate your validated antibody with our kit-provided linker protein. Add it to the Luni where the other end of the linker interacts with the anchor antibodies patterned on the Luni surface, and then you can treat it like any of our off-the-shelf Luni kits. Now you have custom EV analysis ready to go with any antibody you choose. The Leprechaun analysis software makes looking at the details of your EVs a breeze. All titers, sizes, and subpopulation analyses are all done automatically to let you dive right into the results. You can easily access all the fluorescence and IM images to see where the data came from. From here, all results are easily exportable into configurable PDF and Excel formats to let you export any or all of the data you'll need for further analysis or sample comparison. For those of you that are familiar with the analysis software from the ExoView systems, here's where we made a lot of advancements to speed up analysis and make it more consistent. The software now has auto-set fluorescent cutoffs with manual override if desired, and auto-analysis of subpopulations. You don't have to build them yourselves anymore. There's a continuously growing body of literature cementing that this technology is established in the EV field. There are over 500 EV-focused publications utilizing either Leprechaun or its predecessor, the ExoView. If you want to dive into the literature, be sure to include search terms for ExoView and SP-Iris to get the most comprehensive list. We're also happy to help in the search. We have a repository of example publications on our website that can help show how Leprechaun has already made an impact in your specific field. To bring this whole message home in one summary, Leprechaun is the only tool that fully biophysically characterizes your EVs. In a single assay, Leprechaun can provide EV concentration, size down to 35 nanometers, and analyze expression of up to 4 markers per EV. So you find out everything you need to know about your sample from a single assay. Even better, it does it on a single particle basis without the need to purify your sample beforehand. And of course, you'll be able to show off all of that real gold that you've been able to find with Leprechaun. I want to sincerely thank all of you for joining the webinar today, and with that, I will entertain any of the questions that you may have. Thank you.
Great. Thank you very much, Brian. I'm sure everyone here will agree that that was a fantastic and incredibly insightful talk, and we appreciate you sharing your expertise with us today. I'm now going to move into the Q&A session. To submit a question to Brian, please enter your question into the box to the side of the live stream player and click send. We will answer as many questions as possible during the time available. I can see we already have some questions submitted, so I'll move straight into these now.
Great.
All right, the first question we have from our audience is, what are the risks of isolating EVs prior to characterization?
Yeah, good question. So a lot of other techniques require isolating EVs before you go into characterizing them. If you're looking for a rare subpopulation, you run the risk of either separating that population from everything else. So you risk losing those rare biomarkers. Whether or not that is an EV that tends to be a little bit larger or smaller. Different isolation techniques tend to separate by size. So if you have a rare biomarker or a rare EV that is a different size, you tend to risk losing those. The other issue is, different isolation techniques can be, hard on the EVs themselves. So you can actually, destroy the structural integrity of them during that isolation technique, whether it's through centrifugation or through size exclusion chromatography. So, really being able to run the samples crude without having to isolate the EVs really maintains that structural integrity, as well as making sure that you have all of those different EVs still in the sample.
Great, thank you. And our audience would also like to know, what are some of the limitations of NTA alone for EV characterization?
Sure. Yeah, I think NTA is one of those techniques that's really a gold standard in the field. Everybody uses it. It's really good for determining titer, but when you're getting more information about your EVs, trying to get more structural information, trying to look for different biomarkers, get that expression profile, that's where NTA really falls short, right? So having another technique that actually looks at both sizing and phenotype analysis together is a big benefit. Yeah, so NTA, really the biggest issue there is you get a lot of false positives, right? So you're confirming samples or particles that are around the size of EVs, and it'll consider those EVs as well. So that might inaccurately determine your titer that way.
Great, thank you for expanding on that a bit. And can you also talk about what some of the main differences are between Leprechaun and ExoView?
Mm-hmm. Yeah, so like I said in the talk, we made a lot of changes to that ExoView platform when we released Leprechaun. One of the big differences is the size profile, and the sizing that we can measure is now down to 35 nanometers. There are a lot of EVs that are in that 35 to 50 nanometer range, so we get much more accurate titer determinations by being able to measure down to 35 nanometers now. The other difference, one thing that I talked about, was that pan-tetra Luni that we have. That Luni configuration is only compatible with Leprechaun, so if you want to be able to accurately determine EV titer using the pan-tetra Lunis, that will be something you can only do on Leprechaun and not on the ExoView systems. Those are the two biggest main differences.
Great, thank you so much. And we also had a question on whether or not Leprechaun can be used for analysis of samples other than EVs.
Yes, yeah. So Leprechaun is also used for characterizing viral vectors. We have off-the-shelf kits for characterizing lentivirus, but we also have customers who are regularly using it for different retrovirus or different retroviral vectors, things like that. So like I talked about when we have the flex kits, right? So the ability to conjugate your own capture antibodies to the surface. So as long as your particles are in that 35 to 200 nanometer size range, absolutely, we can characterize them.
Fantastic, thank you. And where can Leprechaun be utilized during EV manufacturing?
Yeah, so one of the big benefits of Leprechaun is the ability to run crude samples, like I talked about several times, right? So anything from a clarified lysate, which is certainly a crude sample that has a lot of those interfering other molecules in there. We can absolutely use those because we are specifically capturing EVs out of solution. So one reason I really like Leprechaun for manufacturing and optimization is you can look at samples before and after a specific step in that manufacturing workflow, and you can see whether or not you actually enriched for EVs, whether you were just concentrating everything that was around the same size as EVs, and really dial in and optimize that optimization process.
Great, thank you. We've had a question from the audience regarding the mechanism of cargo labeling. Can you expand on what that is?
Yeah, absolutely. So the way that we look at cargo is by utilizing the kit-provided reagents for both fixing and then permeabilizing the EVs once they are on the surface. Once that's done, you can then use either fluorescent antibodies or, for example, a fluorescent nucleic acid stain, something like that, to be able to look at whether or not you have a nucleic acid cargo on the inside. So either fluorescent antibodies or a fluorescently labeled nucleic acid stain. Those are both commonly used for looking at different types of cargo.
Great, thank you. And what is the effect of permeabilization on EV integrity?
Yeah, good question. So we have a fixing step before we do permeabilization, and one of the kind of baseline experiments that we did was basically to confirm that the size of those EVs was not changing after doing fixing and permeabilization. So what we could show is basically even after that fix and perm step, we still have structural integrity of those EVs. So since they're fixed before permeabilization, we're not breaking them apart during that permeabilization step.
Great, thank you so much. We've had our next question is part comment and part question, which reads, having used Leprechaun for a short while, my question revolves around the fluorescent spots inclusion in the final data. How and why the software determines which are included and excluded from the final analysis? Are you able to expand on why that would be?
Yeah, absolutely. That's a great question, and good to hear from Leprechaun users that are out there that joined today too. So one of the QC steps that the software automatically does is to be able to look at the integrity of each spot and whether or not you had consistency of signal across a spot, whether it was circular, whether or not your blue area of interest, you should be familiar with that if you've used the Leprechaun software before, whether or not that is encompassing over the entire spot. So basically, we're able to perform QC on those to determine which spots are going to give you accurate, reproducible, consistent data. And also keep in mind for every one of those, you can override what it does. So if the software automatically, kicks out a spot that it determines is not acceptable, you can go back and you can turn that back on if you'd like to. So you do have full control over that. But I do utilize that automatic, rejection of samples that are not acceptable for analysis quite well.
Fantastic. Thank you so much for your help with that. We've also received a question that reads, can we characterize markers and payloads together? Will we be able to go beyond 4 markers in total?
Good question. Absolutely. You can look at, well, to the first part of that, you can look at, both cargo and surface markers at the same time. Basically, our fluorescence labeling, what happens with that, after we capture our EVs on the surface, we wash away anything that's non-specifically bound. Then we do fixing and permeabilization of those samples if you want to be able to look at cargo. After that, we have just a single fluorescence labeling step. So you basically pull together all of your fluorescence labels and put those in together. And so yeah, you're able to look at both surface markers and cargo at the same time. To the second part of the question, being able to look at more than 4 markers at the same time. We're limited to those 3 channels of fluorescence and then the 1 capture antibody on the surface. So for a specific, label, or for a specific particle, you're really only able to look at 4 different channels at the same time. That being said, a lot of people will run their samples on multiple Lunis and then just switch out those fluorescent markers for different ones. And that way you can characterize with, different combinations of either tetraspanins, your biomarker, or different cargoes inside as well.
Great, thank you so much. We have another technical question from the audience asking, can you please clarify whether size and counts are measured only for tetraspanin positive EVs, or does it include all kinds of EVs?
Yes, so on our off-the-shelf kits that have tetraspanin capture on the surface, we are only pulling down EVs that contain tetraspanin. Now, if there is another marker that you'd like to look at, something that is more common for all EVs, something that's more universal, which I would argue that the tetraspanins really are. But if there was another marker that you wanted to use for capture, absolutely, you have the ability to look at, conjugating your own antibody to the surface and utilizing any other surface marker for capture of those EVs down on the surface.
Great, thank you. We actually have another technical question wondering if you can only use CD63 labeling or if you can use other tetraspanins for this project.
Yes, absolutely. You can use other tetraspanins. So, depending on the kit that you have, we have capture antibodies specifically for each of those individual tetraspanins, so CD9, 63, and 81. And then we have the fluorescent antibodies for, counterstaining all of those captured EVs with CD9, 63, and 81 in different fluorescent channels. That being said, you certainly have the ability to add, your own fluorescent markers there as well. So if you have a fluorescent antibody for a different marker, a different tetraspanin, you can certainly use that as well. So I know, for example, we have a lot of people that are working with plasma, and we have a specific plasma kit that also has CD41 capture on the surface as well. So, we leave it open to, different tetraspanins, and then you can further customize that yourself.
Fantastic, thank you. Going in a bit of a different direction, one of our audience members was wondering if you have tested milk EVs like blood.
Yes, absolutely. Yeah, we've looked at milk EVs and characterized subpopulations within those a few different times. Yeah, if you'd like to, you can reach out to me separately. We have a couple publications that we've done with that where we work with some of our different collaborators, people who have our instruments, and I can definitely share some examples of that.
Great, thank you. And I will note for the audience, we will be posting contact information as well at the end of the webinar, so stay tuned for that for any further questions there. The next question reads, does the protein corona pose challenges to the characterization using Leprechaun?
I'm not exactly sure. I'm still figuring out how to answer them. That might be a question that, if you want to reach out to me and we can, dive into that a little bit deeper one-on-one. Yeah, in the past, I'm not familiar with, protein corona having an effect on any, EV characterization, but happy to discuss that further.
Absolutely, thank you. And one more question regarding how do you calibrate signal intensity to account for variability in epitope accessibility or antibody binding efficiency across vesicles?
Good question. Yeah, so we use a fluorescent cutoff for each one of our individual samples. right? So for each individual binding spot, or each individual Luni, we have a fluorescence cutoff that we use. And that will account for basically what our background fluorescence signal is compared to what was actually on our spots of interest. So comparing our isotype control to our actual capture spots. So that is tailored specifically for each individual sample and will basically tailor to the fluorescence that you would expect to see for each one of those individual samples.
All right, fantastic. I think we have time for just one more question. So we'll go with this audience question, which is, when we order kits, do we order tetraspanins or cargo labeling or separate cargo labeling?
Sure, and I'm not sure if you have the ability to access our web store yet. If not, you can go ahead and reach out to me and I can make sure that you get access to it. But we recently launched a web store that allows you to look at what kits and what reagents we have available. Basically, you're going to choose the kit based off of what type of sample that you have. For example, we have the pan-tetra kits, of course, and then we have also kits specific for some mouse samples. We have some specific for plasma, that kind of thing. So once you pick your kit, some of them will have optional fluorescent antibodies. So for example, like the pan-tetra kits, you can also add the fluorescent antibodies if you want to. We made those optional just so if somebody wants to determine titer, they can do that without having to pay for the fluorescent antibodies. And then the final thing is, whether or not you want to look for cargo, we have a cargo upgrade, which is available as well. So if you want to be able to look for cargo, that's basically the fixing and permeabilization reagents that you can buy separately to be able to fix and perm your samples to look for cargo.
Fantastic. Well, thank you so much for joining us today, Brian. Unfortunately, that's all we have time for. However, any questions that we didn't have time to get to today will be answered offline as soon as possible. The webinar will now be made available on demand, and to all of our viewers, you can access the recording using the same link you used today. We'll make sure any questions submitted on demand are passed along to the speaker. A certificate of attendance will also be sent via email to everyone who attended today's session. That just leaves me to say thanks again to Brian Haldeman and to everyone in attendance. We'll see you again soon.